So thanks to David and David. I'm Mike Yaffe from the Koch Institute, and I've been charged with telling you a little bit in about 10 minutes or so about systems biology. So I guess the first place to start is what is systems biology, and I'm going to do the same thing that the, I believe it was the Earl Warren Court said about pornography, which is I may not be able to define it, but I know it when I see it. So the type of systems biology that we're particularly interested in is understanding signaling in a variety of tumor types. I'm going to illustrate it with lung cancer, but we're particularly interested in applying systems biology to combination chemotherapy. That is, trying to integrate a variety of pathways. Is there a marker here? Yeah. Great. Trying to integrate a variety of pathways. I guess it's better to just not use it. Pathways involved in DNA damage, metabolism, cytokine signaling, and growth factors to come up with better ways to use combination chemotherapy. Now, there are three premises that I want to use to try to motivate why we want to study things from a systems biology point of view. The first is to remind you that all the monotherapies that are out there for treating cancer are not going to cure the disease. ALK inhibitors, EGFR inhibitors, RAF inhibitors, they don't cure the disease. You get remission for a prolonged period of time. They ultimately all recur. The second thing I want to tell you is that most forms of combination chemotherapy that we use in the clinic right now, CHOP for leukemia and lymphoma, they're not synergistic. Believe it or not, the combination of drugs doesn't work synergistically. What happens is you target heterogeneity within the tumor cell population, but we're not making optimal use of the drugs themselves. And the point I want to leave you with is that systems biology is the key to coming up with effective synergistic combination chemotherapy or chemoradiation therapy to eradicate tumors by understanding how it is that signals within tumor cells are propagated and understood. Okay. A quote from Craig Venter that just sort of illustrates my view of systems biology. You'll hear probably a slightly different version of systems biology from Mark, who's going to speak after me. And what Craig Venter said was, you can't study one protein at a time. I would expand to say you can't study one signaling pathway at a time. You have to look at how signaling pathways crosstalk and interact with each other. And so to give you just a general flavor of what we do at MIT and then one specific cancer example, we think of biological circuits much like electronic circuits. Instead of wires connecting electronic components, we think of post-translational modifications like phosphorylation or acetylation or methylation as basically being the wires that carry the current of signaling. If you want to understand how a complicated circuit works, you can't sit there and measure the voltage or the current at one point. You have to measure the voltage or current or whatever at many points in time, and that's what we would like to do. We would like to measure information flow through signaling networks using metaphorical meters, and I'll show you the meters that we use. We've got a variety of technologies that let us measure activity in many pathways all at the same time, and then use that information in combination with multiple agent treatment of tumor cells to understand how best this can work. And the fundamental premise, of course, is that if we can do this, if we can give the cells some sort of a cue and then measure as many signals as we can and then look at the response, we can build mathematical models in which the responses are some function of the signals. So the responses here are some function of the signals, and what we would like to do is be able to measure as many of the responses and signals as we can in order to derive this information processing algorithm, because it's going to show us where the nodes are in the network that we ought to be targeting to cure cancer. In order to do this, you have to turn to mathematics. There's no other language that can capture this kind of complex information. And I've shown you here some mathematics that looks a little complicated. The types of mathematics that you use, I'll show you in a moment, don't have to be this complicated, and they're all relatively straightforward things. I trained it by way of background. I trained as an engineer, and then I was an MD, PhD, and trained completely in general surgery. So I try to bridge the engineering and clinical medicine disciplines together. And if I can do this kind of math, everybody here can too. So the types of math that are available depend on the question that you're asking and how much data you've got. So if you're asking a very specific question and you have a lot of mechanistic data, you want to use things like differential equations. If you want to simply map relationships between activities and pathways and responses, you can use things like regression and clustering. And I'm showing you here all the different approaches that we typically use at MIT. And as I said, you can start from very abstract models using things like PLSR and clustering. You can go from very specific mechanistic models with differential equations and everything in between in order to get insight into this. There are 4 M's that we think about when we talk about systems biology at MIT, and that is mine, model, modify, and measure. And so we start by doing the experiments we want to do. At the moment, it's often in tissue culture, but we're doing more and more of these now in tumor models. We'd love to take this to patient samples if you guys are interested in working with us. We mine that information, we build models, and then we modify the experiments and test the models. The most important thing that I would say here is that it starts with measurements. You can't, in my book, you can't do systems biology with just a pad and paper. You have to start with measurements. And the models have to be insightful and predictive. They should tell you biomarkers personalized treatments and new targets. Let me give you one very quick example of how we've used this, in this case for triple-negative breast cancer. We were particularly interested in trying to optimize combination chemotherapy for breast cancer, and we wanted to focus on one type of breast cancer, triple-negative breast cancer, because it tends to affect young women. Many of you know this better than I. And it's the type of breast cancer that has the worst overall prognosis. And what we wanted to do was to ask, could we combine the standard ways that we damage DNA using chemotherapy or radiation with specific pathway inhibitors that targeted some of the pathways that you see here? But what made our approach different was thinking about networks and systems. We realized maybe the thing to do wasn't to just combine the two drugs at the same time, but it might be to give one drug and wait and then give another drug, or give the first drug and then wait and give the second drug, so that we allowed networks to be dynamic in cells and to rewire. And we did this experiment, and the story I'll tell you very quickly is, I think, a new role for two old drugs, doxorubicin and erlotinib, in treating triple-negative breast cancer. Well, as many of you know, there have been clinical trials where patients are given doxorubicin and erlotinib together, and the trials have been pretty disappointing, as shown here, for example, in the survival of cells treated in culture with erlotinib or doxorubicin or the combination treatment. And you can see you get a little bit of increased cell death if you mix the drugs together, but not a lot. The surprise was that if we added the erlotinib and we just waited between 4 and 24 hours, we could increase our ability of doxorubicin to kill these cells by 500%. If we reversed the order, we gave the doxorubicin first and then erlotinib, we got less death. So clearly, two drugs that are already in the clinic could be used much more effectively if we simply thought about this from a systems biology point of view. It's very tumor type specific. This is wherever you see an arrow, like E arrow D, means pre-treat with erlotinib, then treat with doxorubicin, or vice versa. You see it works really well in triple-negative breast cancers. You get increased cell death. You actually get less cell death if you do the same treatment with HER2 overexpressing breast cancers. So it depends on the tumor type. Okay? Now, obviously, we're signalers. We wanted to understand how this worked, and so we made a very simplified model of some of the pathways that we thought were involved. Things that would connect doxorubicin-induced DNA damage with EGFR-mediated signaling. But the nice thing about this is we can measure very accurately everything you see here in white, and now about a third of the things that you see here in gray. And so what Mike Lee did was he made measurements, each in 3 different cell types, of all of these different pathways I've shown you at 11 different time points in triplicate. And we can do this for basically almost any pathway. And we can do this probably, I think, in real tumor cells. I mean, in real patient tumor samples. And in addition, we can do this dynamically. So what you're seeing here isn't just a single measurement. It's a time course measurement of the activity of all of these different signals, the details of which aren't particularly important for this brief talk. In addition, we did this using a variety of things. We did this using micro-Western blotting. We did this using reverse phase protein arrays. But we were able to build this large data set of signaling. We did the same thing for responses. We could measure, for example, where cells were in the cell cycle. We could look at whether they were undergoing autophagy. We could measure quantitatively by FACS how many of the cells were dying. And we could look at their overall viability by looking at things like ATP content. And now we have these 2 big data sets. We've got a signaling data set, we've got a response data set, and we can use a variety of mathematical approaches that I don't have time to go through in order to try to model this. The advantage of doing this is you can build a model. Here's a model that tells us in those triple-negative breast cancer cells how much apoptosis we would predict for a particular treatment and how much we measured. And you can see that the correlation's fantastic. The best thing about the model is you can now ask the model, why? What's so special about this pre-treatment effect for combination chemotherapy? And can I use this to identify a biomarker? Can I use this to identify, for example, a response marker? And what the model said was that it was cleaved caspase 8 that was the key determinant in whether or not pre-treatment was going to kill the tumor cells or not. It said that, in fact, in a subset of triple-negative breast cancer cells, when you gave erlotinib and you waited, and then you gave doxorubicin, you saw an increase in caspase 8 activity that was responsible for the cell death. You can test the model. The model predicts that if you get rid of caspase 8 with RNAi, the effect of the pre-treatment with erlotinib will be reduced. This is what's predicted. We knocked it down. This is what we measured. It beautifully recapitulated what the systems biology model said would happen. So now we have a mechanism for this. We have a response marker. Does it work in vivo? These are mouse xenografts with triple-negative breast cancer cells. If you treat these mice with 1 dose of doxorubicin, I think you can see here the tumor gets smaller and then it recurs. If you treat the mice with doxorubicin and erlotinib together, again, you get a slightly bigger reduction in the size of the tumor, but again, it recurs. But if you do this pre-treatment, you give the mice 1 dose of erlotinib, wait 24 hours, give them 1 dose of doxorubicin. At least over the time course of this experiment, not only did the tumor shrink, but they did not recur. Furthermore, we were able to identify which subset of triple-negative breast cancer cells are likely to respond to this kind of treatment. What I'm showing you here, I'm going to ask you to focus, if you would, just on this panel right here, where I'm showing you the response of these different tumor cell lines, all triple-negative breast cancer tumor cell lines, to doxorubicin alone, shaded in one color because it's the same response. And now if you focus on this panel, which is the pretreatment effect, you can see I can rank the cells from those that show the most sensitivity with pretreatment to those that show the least sensitivity. The interesting thing is there's no correlation with EGFR expression. So all the people in the world, all the Joe Grays in the world who are telling you you're going to measure EGFR expression and predict who's going to respond to erlotinib, they're not right. Doesn't work. Because it's not the levels of EGFR that tell whether the pretreatment works. In fact, it's the basal activity of EGFR. If we simply ask how much are those breast cancer cells signaling through the EGF receptor pathway in the basal state, you can see there's a beautiful correlation here between the activity of EGFR in the basal state and whether the cells respond to the pretreatment, and that correlates exactly with whether they activate cleaved caspase 8. So you see what this gives us is a biomarker for which patients to treat. It should be the patients whose tumors have high levels of phospho-EGFR. And we have a biomarker for response. We could biopsy the tumor after treatment and ask, did the tumor activate caspase 8? And if so, that means our treatment's likely to work. So the model that emerged from the systems approach was that if you chronically treated cells with erlotinib, you could suppress an oncogene signature and unveil a caspase 8 dependent pathway that was likely to increase tumor cell death. Last slide I want to show you is this something unique about triple-negative breast cancer cells. I'm showing you here 3 different lung cancer cell lines. In fact, the results were even more dramatic. In these 2 cell lines, I hope you can see that if we pre-treat with erlotinib and then give doxorubicin, we see this tremendous increase in apoptotic cell death compared to if we give the drugs together. So I think that this general approach of being able to take drugs that are already out there in the clinic and use systems biology to figure out how best to combine them is likely to gain a lot of traction. So what I've told you very quickly is that I've shown you an example of how systems biology can work for combination chemotherapy. We would love to do this together with some of you, with those of you that are interested in helping us make the leap to the clinic, look at clinical samples, look at for example, at accumulated data that you may already have on patients. We can even do some of this retrospectively on patient samples. And I've shown you here an example of how, for one case, the EGF receptor and the DNA damage signaling pathway crosstalk. I've shown you how combination chemotherapy can use more effectively if you use one drugs to what we call dynamically rewire the signaling pathways to make the cells now more sensitive to a second treatment. I think this has not nearly been explored to the extent that it should be. And I've tried to tell you that this concept of dynamic rewiring can shift the focus from having to identify new drugs to basically using the drugs we already have combined with some of the drug delivery approaches that you heard about earlier to create, for example, new ways to use old drugs more effectively. I would argue that we may already have all the drugs we need to cure cancer. We just don't know how to use them correctly together, and I think that's where systems biology is going to take us. Key point here is we have to test these systems about dynamic network modulation and optimizing combination chemotherapies in the clinic. And that's where I'm hoping some of you will be interested, and if you are, please drop me an email. I'll stop there. Thank you.